Good morning, good afternoon, depending on where you are. Yeah, it's a pleasure to have you here and welcome you to one of the events of the eBrains webinar series. There are multiple webinars. What we are doing from the perspective of eBrains is we want to demonstrate how we can make it easier to lower the threshold, how to use eBrains in particular, manipulate data, tools, and the models. And there is a series of this type of webinars. There was one on brain atlases, another one is coming up. And today is dedicated to multiscale brain simulations in eBrains, from the neuron to the patient. So you see it's already across multiple scales. And this will be animated by Professor Egidio D'Angelo, who's here with us from the University of Pavia. And myself, I'm from the Aix-Marseille University, and in eBrains, I also serve as a Chief Science Officer. So this webinar here, so wonderful that you're here. It will be also recorded, and the recording will be made available later on the website. Yeah. Today's agenda, the way we have envisioned to spend this 1 hour that has been allocated to us, is as follows. I say a few words of setting your mindset, a framework of how to see this today's webinar in the context of eBrains activity. And then Egidio will guide us through the use and some very illustrative use cases in using microscopic and mesoscopic modeling. Then I will take over and go all the way to macroscopic modeling and how these models can actually be used in the context of eBrains applications specifically to clinics. And then afterwards, we will still have, I hope so, a few minutes for questions and answers. For the Q&A, please, the way we have envisioned to do this is we have in the background Evan and Ocean from the eBrains team that accompany us. Simply send the questions in the chat. And like this, Evan will collect them and he will then pass them on to me so we can actually go through them. Yeah. And then, if possible, also mention whether there is a question specifically targeting Egidio or me. We will also try to answer it, both of us. Huh? So let's get started. Yeah? For this particular webinar today, I'd like to first of all start with a quote from Sidney Brenner that I simply will read out to you. We're drowning in a sea of data and starving for knowledge. The biological sciences have exploded, largely through our unprecedented power to accumulate descriptive facts. We need to turn data into knowledge, and we need a framework to do it. And this is actually very important. This particular framework that we need in order to do this, how can we do that actually? Yeah, data is not knowledge. Models by themselves need to be connected to the data. So the way I tried to paraphrase it here and there, I took inspiration from Mackamer, Darden, and Cravers. This is a group of philosophers that have written an impactful paper in 2000 thinking about mechanisms. There they used an ontological definition of a mechanism. So asking the question of what is a mechanism? But I paraphrased it in the following sense, namely in an epistemological sense about how to actually understand, how to generate this type of understanding. And that is through explanation, thus subsequent understanding. This involves revealing the productive relation between causal activities linked to their constituent entities. And that provides us with a recipe, yeah, to generate this type of understanding, pretty much like what Sidney Brenner was asking for. Data is not the understanding per se. We need a roadmap to this. We need to identify the constituent entities. We need to have a causal hypothesis. We need to formalize the causal activities, and then we need to put them into a productive relationship in order to generate this type of understanding. And this is at the end of the day what we are after as scientists. This putting in a productive relationship, the revelation thereof, this is into operation. And this is exactly what we are trying to do in eBrains. And one mechanism, one vehicle to do so is digital twins. And some years ago, we have written as a community a paper in imaging neurosciences that's down here, the coming decade of digital brain research, a vision for neuroscience at the intersection of technology and computing. So this paper was in the making for several years. It was available on Zenodo, and people were able to contribute to this. Community was invited to support the paper or to criticize it or to also contribute as a writer. And the idea was to define the next coming decade, lay out the path towards this. And the digital twin plays a very important role here. And I put it in bold here. Digital twin is constrained by data, that provides simulation data to guide choices and anticipate their consequences. So a digital twin is thus a copy in the practical sense, that it is defined on a particular level and is essentially intended to be useful. And in our hands, and that's what we are going to talk about today, is a digital twin is organized across multiple scales. And what it is a representation of our knowledge, a formalization of our knowledge. And once we put it into a mathematical, symbolic, algorithmic form, then this knowledge becomes a model. And this is what we are representing in digital form here in the virtual space. What we are injecting in there are data. We fuse it with empirical data. And then we have to juxtapose, put in relation the empirical input, the data, and the models, and identify the underlying causes. So this is a realization of the cause-effect implementation. When we have this, we can actually use this in two ways. Either we link it to the simulator, and then these models can actually, in an autonomous fashion, generate brain activity themselves and allow us to interrogate it. So that's like a flight simulator for an airplane. Here it is a brain simulator that simulates this fusion of data, empirical space, and our knowledge represented as a model. This is what is happening in the virtual space. Autonomous simulation. And the other form of usage is you can use it actually as a template to start to make it subject specific or patient specific in the context of estimating certain parameters. We call this inference. You have underlying causes that you would like to estimate, and you can do this specifically for the data set that comes in. This will be addressed in the context of inference and model inversion. This is how we operate. In eBrains, we give you actually the capacity to do this in this interoperable fashion. So I just use this image here as a full brain network, but what we offer in eBrains is actually we have representations of digital twins across multiple scales, and the scales are often also connected. We speak about this in more detail. Microscopic scale, where we have detailed cellular, subcellular representations, allowing us to build, here in this case, columns of multiple neurons, or microcircuits, mini networks, going up all the way to the whole brain network level. In order to explore these models, we need an underlying compute structure. This is being provided. This is already a huge leap forward because these models are linked to simulators. The simulators run on the hardware, providing storage and the compute power needs. Something that is very important for the experts here, or for those of you who have already worked with models, is they need to be parameterized. You can be so off in parameter space. If you don't know where you are, the models may generate completely meaningless results. So a parameterization, tools for parameterization are very important. And what we are have developed over the past years and is now available are these off-the-shelf models that are well parameterized, that provide you with a good starting point, and this lowers the threshold for entry into the eBrains ecosystem, allowing you to use these models. And then, very importantly, we link them to data. Data come from different sources. And this is already another way of interoperation, because the data come across different scales, sometimes from different species. Yeah, because not all the data are accessible for the human. Sometimes they need to be informed coming from other species. So there are multi-scale data often coming from different laboratories, either for a single individual or for cohorts, often ex vivo, yeah, coming of different nature. Here you have a connectome. So there we're talking about topology. Here we're talking about many, many geometric representations, yeah, or patient-specific data. If you're interested in clinical patients, this is patient-specific data. So what we do in eBrains, we are interoperating this. And for this, I really invite you to go to the website and actually enter through the portal and look at the sequence of services. And the goal is essentially to lower the threshold and to enable you to generate outputs, either medical output, multi-scale, like here as an example, medication, or even robotics, technological situation. That's what we do in eBrains. This is the vision behind the interoperation behind that. The way we have broken it up in the eBrains structure is in 6 categories. Brain atlases that provide the anchors for navigation within the eBrain structure. Yeah, medical analytics, so multiple platforms dedicated to medical applications. Modeling and simulation. This is where we will show you some examples today of how to connect models to the simulation engines. Yeah, data and knowledge. Yeah, organize the knowledge following FAIR principles and link them to the other tools. All this within a collaborative platform where we allow you, or the platform allows you to enter with your colleagues and work together with the projects, and all supported by the compute infrastructure. That's what I wanted to share with you, just as a perspective. Have this mindset. I invite you, really, I encourage you, go to the website, because our website in eBrains is our entry portal into the infrastructure. It's easy to get an account. And the 6 categories that I just showed you here, when you go onto data, tools, and models, this is what you find, and it's declined into many different categories.
So thank you, Viktor, for the introduction. I think the main issues have been clarified. So I have an easier life. I will move more quickly through the foundations. And then I will proceed by showing examples and principles, yeah, that I think is the most interesting thing to do now, to allow you to come smoothly into the eBrains ecosystem. We perform in eBrains multiscale brain modeling. Multiscale brain modeling is generated because of the multiscale organization of the brain. So we needed to take into consideration different level of organization and complexity of the brain. And we needed to jump across the scales and connect the scales. Connecting scales is a critical problem in life science and in physics and in chemistry. It's not always easy to fix. And the way that we're following is to generate models that allow to predict what happens at the different levels of the scale. So inherently, our models must be multiscale. This goes hand in hand with the capacity of recording biological and phenomena on different scales, which means that we have complex data sets that are organized on different scales themselves. So for example, we're recording from single neurons, from multiplicities of neurons, and from brain areas where we don't see the neurons, but we see the collective activity of neurons. These neurons are organized at the moment in eBrains in what is called the knowledge graph. The knowledge graph is a database that is organized and curated in order to allow access to these data on different scales. In In a work that was done by Viktor and me a couple of years ago, we have formalized the problem of generating models across scales by considering that these models can go in two opposite directions. One direction is from single neurons and what we call the elementary causes to build up ensemble signals, which means crossing the scale upstream from the micro to the macro scale. The other way is to go from the macro scale toward the micro scale, which is much more complicated, implies inversion of models. And this is an aspect that Viktor will consider in detail in his talk after me. So what I will consider now is the direct problem, which means building models starting from the elements, from the neurons, and even more from the molecules that populate the membrane and the cytoplasm of the neurons. In order to process the models on a multi-level scale, there are several workflows and streams. The one that I will speak more now is this one, starting from cells, generating neurons and microcircuit models. Viktor will talk about the brain and starting from the brain to extract information about the mechanisms that work inside the brain. A parallel pathway is about the neurorobots in which we can embed sort of virtual brains or microcircuits and that we can use to interpret the activity of neurons and circuits in terms of behavior. I will give some touch on it at the end of my presentation. So first column, second column, and third column. Yeah. Now move from molecules to neurons and networks. This is the basis. This is the physics of the system that takes into consideration the membrane equation, electrodynamic equations, and oxygen-Huxley gating equations, or similar. There are also stochastic models for molecular transition from one state to another, the Markov chains, that can be embedded into the models. I will not go into the details of this description, but just to consider that we work with systems of differential equation that in the end are capable of generating the strongly nonlinear dynamics of membrane voltage changes in neurons. So by leveraging these equations, we can generate the models of neurons. Now I show more in detail what we can do. This is an example in which a neuron has been reconstructed starting from its own morphology, has been divided into multiple electrical compartments, which have been connected to synapses. And finally, this model is run into the simulator called the Neuron. Neuron is available on eBrains. Other apps allow to extract features from the action potentials and the membrane potential changes of the neuron. And other apps allow to optimize the parameters that remain unknown, mainly the maximum ionic conductances in the different compartments of the neuron. So there are parameters that are unknown. These parameters can be extracted using specific apps in eBrains. And then we can generate a parameterized version of the neuron that can run and be embedded into networks. The embedding into networks is a complex problem, mathematically speaking, which requires special tools like the Brain Scaffold Builder that is also on eBrains platforms. So now, at the end of all the process, we can generate models of neurons and microcircuits, and these models can simulate the activity of the different components of the network. An example is shown here. This is an example of a neuron in which the membrane potential changes are represented in the different compartments. And then we can visualize calcium changes in the cytoplasm, AMPA currents, and NMDA currents, and GABA currents in the different synapses. There are visualization tools that allow to do the job. So the data can be inserted into these tools, and these tools provide the visualization of neuronal activity. If we move forward to the network level, this is the visualization of what a network is doing. There are many neurons, like the one that you have seen before, that are running and connecting together, generating what we call the spatio-temporal dynamics of microcircuit activity. So this is a visualization of what a network of the brain is doing. This is a part of the cortex of the brain. Yeah. Now, how do we use the models? The first step that I told you is how to build them up. Now, how to use them. To use the model, we need experimental observations, we need hypotheses, and we need models, which are differential models. The example is taken from gravitation, the Newton's law of motion. But the problem is exactly the same. We have dynamic systems in which we have systems of differential equations that describe motion of some components of the system, of some entity into the system, and they allow to predict the existence of something that is unknown. Because if we knew everything, the model would be useless, would be just a reproduction. We needed to extract from the model missing knowledge. As Viktor explained, we needed the models to understand. And therefore, our model generate predictions. The example here is about Planet 9. Now is the famous problem of astronomy. But now turn it into problems of neurons. Neurons are based on observations. Follow this example from follow the arrows. Information is allows to build models. We jump here. The models doesn't generate all what we want to understand about the neuron. There is something missing. And therefore, we introduce into the model new functionalities. These functionalities predicted the localization and the existence of new ionic channels that were not present in the initial observations. We have discovered Planet 9 in a way, just to make reference to the example that I showed before. Planet 9 allows to generate a better model, and we use it again. We make another prediction, and then we discover other properties of the axons, of the channels, of the electro-responsive mechanism of the neurons. And finally, we use the model again, and then there is yet another channel. I think the story is not finished. It will go on again. So these models have a strong predictive power, are transforming what is a sort of serendipic and curiosity-driven science into a model-based science. Now, this is another example that is brought up to the level of a network, in which the properties of a network generate map of activity. This map of activity are reproduced by a network model, and the network model predicts how plasticity deformates, changes the map, supporting one of the main theories of the brain, that is the Marm's motor learning theory. So basically, the models can be used to support theories. It is true that these models are computational, but it doesn't mean that they only make computation. They can support theory. Another aspect is shown here. We have reversed the paradigm. We started from model prediction. This is exactly the model that you have seen running in the movie, the model of a Golgi neuron of the cerebellum. This model, run in appropriate ways, predicts the existence of spike time independent plasticity at certain synapses. That was completely unknown. Nobody knew about that. And then we started, based on the model, to look for this short spike time independent plasticity, and it is here. It came out exactly as predicted by the model. So this, again, tells you that this model has a strong predictive power. Last but not least, now the models that were previously used only for neurons of rodents, because of the availability of data coming from rodents, now are being used also for human neurons. And in the human neurons, they prove very interesting because they're predicting how these human neurons compute and how these human neurons differ, if they do, from neurons of rodents. This is an example of the Purkinje neurons of the cerebellum, in which the models allowed to estimate the computational capacity, which turns out to be all 1 order of time higher in humans than in rodents. So that's the way that the neuron models and the microcircuit models can be used. Now, we wanted to bring these models into the virtual brains. The virtual brains will be treated by Viktor, but I will show you how we can bring the knowledge coming from biology and passing through the model of reconstruction that I showed you into the virtual brains. The first problem is that we can't bring the models with all the complexity that I told you they are directly into the nodes of a virtual brain. This is why they require too many ordinary differential equations to be solved, and we have to reduce their complexity in computational and mathematical terms. So we can bring down by orders of magnitude in their complexity to the level of a couple of differential equations. It is, in many cases, it will be enough. In other cases, it's not enough. We can reduce them even more and compress these models made of many neurons into compact representations that we call mean field models. It is shown here. These are the models with all the compartments. These are point neuron representations of the models in which we have single point neural models connected together that allow several interesting features to be analyzed. For example, large scale, synaptic plasticity, long-term simulations. Okay. These models can be compressed into what we call the mean field models. So this is the signal on the mesoscale that can be introduced into the virtual brain twin. I want just to give you a flavor of what happens when we follow this procedure. This is an example of a virtual brain simulated using the virtual brain platform, TVB, that will be explained by Viktor, in which the nodes are coming from the workflow that I showed you. Single neuron at high resolution, simplify the neuron, spanking neural network, mean field model, and the mean field model goes into here, into the nodes. Okay. When the model starts to run, the simulation shows sort of electroencephalographic trails in each one of the nodes. What is behind it? The mean field models are allowed a multi-regional organization of the network. For example, this is done for the cerebellum, but other groups have done it for the cerebral cortex in the CNRS in Paris. The different components of a region can be connected together, can be pre-wired, respecting the internal connectivity, respecting the multi-regional organization of the structure, and also allowing to represent differentiation in the regions that compose, for example, the cerebral cortex or the cerebellar cortex. This is an element that is very advantageous in order to generate effective twins. Yeah. The other thing is that we can analyze a different population of neurons independently. For example, this is the case for the cerebellar cortex, where we have granule cells, Golgi cells, molecular layer interneurons, Purkinje cells. They can all be analyzed independently. We can generate maps of activity that are very resemblant of the activity of the brain of subjects. We can extract, for example, cell-specific fMRI signals. We can also extract a cell-specific oscillatory pattern. For example, theta band, gamma band, whatever happened in that particular population of neurons. So the fine grain that we can transport into the virtual brain becomes really high. Eventually, these models make a step ahead compared to virtual brain models that do not use this strategy, but the more simple representation of the nodes. That's the issue that Viktor will expand. Now, very last touch, what happens when we transport the spiking neural networks into robots? This is an example in which the neural network that you have seen running in the movie have been simplified and transported, inserted into the controller of a robot. It can predict the evolution in time and enduring behavior of neuronal discharge. This is an example of the Purkinje neuron of the cerebellum. You see this black line here. The neuron is learning the correlation between events happening during the interaction of the robot with the environment. It is an example of adaptation of the system that is autonomous, is based on rules of synaptic plasticity and on the circuit properties that we have introduced into the controller. So this is a robot with neurons inside. It's a sort of hybrid robot. Okay. I just show you the example of it. This is the robot that is learning to follow a circular orbit with the arm. Okay. After a couple of trials, the robot is doing very well, is performing well. Now you will see what happens when the robot is disturbed, the arm is loaded with a weight. The graph on top shows the distance between the red and blue trajectory, okay? Now, the arm is disturbed by weight and falls down as expected. But what is not completely expected is that the robot autonomously learns to correct the error. You see that the error, which is the distance between the two orbits, the error progressively goes down. This is because the system autonomously learns to correct the error by using the internal circuit, by using the rules, by using the neurons. Okay. All these facilities can be provided by specific apps in EBRAINS. So one can also carry out robotics simulations. In conclusion, multiscale modeling is helping understanding the multiscale and multiparametric world of the brain. And multiscale modeling is the basis to construct the virtual brain models and brain digital twins. At this point, I leave the word to Viktor that will continue the next step.
I'm taking it now across scales. So, and go back to this one particular slide that we started off, yeah, with regard to the organization of digital twins and so the fusing of empirical data and in a digital ecosystem in which we represent the models. And if the data come from an individual's brain imaging data, typically they are on a millimeter scale. Yeah, if you think of MRI, CT, or also centimeter scale, if we think of EG, MEG, also fMRI. Yeah, then we refer to this as virtual brain twins. And this type of virtual brain twins, they can be used for two operations: autonomous simulation and inference. Yeah, in both cases, there are predictions being made, but the virtual brain is being used in two different ways. And this is essentially what I want to unpack here for you, with you. And I wish to emphasize every single operation that we are discussing here is available on the eBrains platform. And there are also many help sites that can help you to navigate through the website, through the portal, in order to understand how to find that. Yeah. So first step for the construction of a virtual brain twin is we start with an individual's brain imaging data, specifically the anatomical data, structural data. Doesn't have to be MRI. It can be also anatomical MRI, diffusion tensor weighted imaging. It can be CT data. Yeah? They enter into a shared ecosystem and are being co-registered. So they are being represented in the same atlas. Yeah. The same applies also for functional imaging data. They enter into there. The electrodes, so the sensor locations, are being also registered in the same space, so it's completely coherent. And then we equip the thus resulting geometrical structure. If you want, you can refer to it as an avatar, as a person-specific avatar. We equip it with mathematics. And this mathematics then renders this twin functional and can be used either for simulations or for inference, so as a causal hypothesis in which the parameters can be estimated specific for the data you enter. This could be seizure propagation data. It can be pathological versus non-pathological data. It can be cognitive data. So this is a process that we refer to as inference. And as Egidio pointed out on this one slide, the parameters are the actual entity that connects data and the models. So for forward simulations, we need to set the parameters. For estimations of like pathophysiological causes, we need to estimate the corresponding parameters. Yeah, and this provides us with the input that is then being fed back to the user. Here, in this case, it's a clinical application. So let me step you through this. When you load in the data, there are workflows, pre-processing workflows that allow you to co-register the data sets in the same space. MRI data, they undergo a reconstruction unfolding in 3D and a parcellization, as you see here. Then they are being co-registered with the diffusion tensor weighted data that undergo tractography and allow you to build the connectome, spread it and define it in three-dimensional space. Then you link it to mathematics, yeah, and the parcellization gives you the various network nodes. But a very important step, and this is the way, one of the major reasons why the virtual brain has become such a strong vehicle for clinical translation. You're in a physical space, which means you can actually represent the sensors. Yeah, like for epilepsy, we do stereotactic EEG to record the activity in the volume, hence stereotactic, of an epileptic patient. And then this is where the physics enters, because the sources are these green cubes here. This is where the activity happens. The dark green spheres, little balloons, are the SEG electrodes that actually sample multiple activities. So there we have Maxwell equations in matter, yeah, that communicate between the sources and the sensors. MEG, same thing. EEG or functional MRI, it's a neurohemodynamic coupling. So this is what we need to deal with. In order to equip this type of spatial configuration, we need to enter mathematics. The mathematics, these are multiple decades of research that enter here, and one means of mathematics that is being used in the virtual brain twin are the mean field models. So these are the models on the network node. In this visualization, it is a different red spheres that correspond to an individual brain region. This one you equip with a neural mass or population equation, and this can be derived in different ways. Egidio spoke about the mean field techniques. There are multiple PIs that have contributed to this, and Alain Destex being a pioneer in this domain, using statistical techniques to describe the activity of a population of neurons on one of these network nodes. Mathematically, it looks this way. Yeah, you have a state vector psi at a location x at a time point t, and this is your neural mass equation, your mean field equation. We are dealing with differential equation, which provides a forward in time evolution of the activity, but it depends on the other locations also. Intracortically, you connect through the gray matter. This is nearest neighbor connectivity. But through the connectome, here you have large-scale projections from X prime to X. These are long, up to 180 millimeters, these fibers. So they undergo a time delay via signal propagation. This equation, that's the virtual brain equation, which is agnostic to start with to the type of activity, how much detail you have inside to architecture here. It depends. This detail is in the population model that you choose. So you choose as a scientist responsibly, build it, and the tools you have in eBrains helping you to build it up. And then it requires quite some numerical mathematical sophistication to solve it over time. This is what the simulators do. It's difficult because you have time delays in there. It's difficult because you have noise in there. It's very difficult because you have noise, time delays, high dimensionality, and nonlinearity in there. This is what the simulators are for and optimized. So that provides you with a little bit of organization. Here's the summary. So what I just told you is structural data and functional data enter into a shared space together with sensors and reconstructions in 3D. I also spoke about the mathematics that we are helping to provide. And we have so-called off-the-shelf models, model templates that lower this threshold for entry that we have optimized for you that you can start working. But of course, as a subsequent step, you will start modifying them to your needs, to your cognitive tasks, to your pathophysiology you wish to address. Yeah. So this is where you manipulate the parameters in there. For this, you have to know how to express the pathophysiology in the model. In multiple sclerosis, it's a time delay through the connectome. They become increased because of the demyelination. In epilepsy, look for the epileptogenic zone. Yeah, these are the activities. So it's you as a scientist that is driving the question, and we have provide you with interfaces that allow you to ask this question. If you want to use the virtual brain twin not for forward simulation, I'll test some hypothesis, but as a template for fitting, yeah, such as to find the epileptogenic zone or find the lesions in your MS patient, you have to perform inference. This is the second layer of operation that is very specific for the virtual brain twin that then leads to diagnosis or identification of intervention strategies. Let me give you examples in the remaining time that I have. Yeah. Virtual brain, what I showed you on this example here, you see you can actually count the nodes. In this case, it's typically 164 nodes, some subcortical areas, left and right hemisphere. You can also go to a high resolution. What we typically do is multiple hundreds of thousands, and that gets us to a resolution of 1 millimeter. This is actually very good because it allows us to use the detailed data that we have in eBrains to reconstruct the cortical surface. Here you see a tessellation there of the triangular structure. This is for the cortex. We take a volumetric approach for the subcortical areas, of course. Yeah. This is an example from Paul Treepkorn in my lab. Dr. Paul Treepkorn, he graduated very recently. Yeah. So taking the hippocampus that is represented here, what you can actually, this is represented down here, you can actually inflate it. Yeah. So from the convolved structure to the inflated structure and then map it on the sphere. That's mostly for better visualization. You have the same color code here that is represented on the sphere. And then you can run simulations. Again, hippocampus, CA1, CA3, CA4 region is here. And here in these areas, what Paul did, he actually took the simulation and he increased the epileptogenicity in the hippocampus and initiated propagation. So seizure activity, and it starts evolving, and then it starts propagating, exploring the geometry of the seizure, of the tissue, propagating into the non-epileptogenic areas, and then stopping. Yeah, you see the classic slowing down, and then reorganization on a more larger scale, slow down, and then we arrive at the seizure end. Yeah, this is actually fairly straightforward without very complex manipulation of the parameters. Yeah, here you have another example. This is a rhinal cortex. Yeah, so we can actually display the tracts that are coming out of the rhinal cortex, intrahemispheric and interhemispheric. Here you have a vertex connectivity strength. You can increase the epileptogenicity in the rhinal cortex and then run this type of stimulation. So the rhinal cortex is here. You will see activity will go up. goes up. And please note, did you see it did not propagate through the tissue? It required the extracortical connectivity. Otherwise, it could not have done this further distance emergence of activity. And then it started reorganizing self, and you have again this propagation. Yeah. So this is a type of work you can do. You can go to the all the way to the a full brain level, which you see here. It's a patient that we reconstructed, and here it becomes important where your SEG electrodes are. Activity propagates through the network. Here, the energy is represented by these spheres, yeah. And as the activity propagates through, and the different electrodes, so you have the A electrode, the B electrode, one is in the amygdala, the other one goes in the hippocampus, and you see the classic seizure organization. Yeah? Propagation through the network. This is a type of data the clinician would see. Through the twin, you have access to the source space. Otherwise, you never have access to the source space. And us as scientists, we can now operate on these two levels. And this type of technology helps you to make the bridge between source space, in which the causes are actually living, but the data we see in the sensor space. Yeah. So that's this is what now you can go in and start changing parameters, perform virtual surgery, start making a resection. What are the consequences? This is a type of question you can use in a forward simulator. Yeah. If you are interested in multi-scale cooperating, what we did in EBRAINS, we developed a technology which we refer to as core simulation, and you can actually run two simulators: the NEST simulator that Egidio referred to, and the TVB with the virtual brain simulator. You can run them simultaneously. Yeah. However, given the complexity, NEST simulates single point neurons. Yeah. So you have to focus on a region of interest. You cannot do the interbrain, at least not in our hands in the EBRAINS platform. Yeah, and very often this is actually not a question we wish to ask at the moment. Yeah, so you can co-simulate them. So here in this example, it's a hippocampus reconstructed in detail with NEST, and the rest, the remainder, you simulate with the virtual brain. This is some data that have been used from Javier de Filipe. And once again, data and workflows and all computational models for this example, which is a beautiful example, is also, just look for it on the website. And Gandolfi et al. In this paper we have published, this has been spearheaded by Michele Migliore, the entire workflow. Anatomical data, electron microscopy, the neurons have been identified in position three-dimensional space. From the images, you can reconstruct the axonal dendritic tree and build probability clouds and then start connecting them. Yeah. And then build the CA1 region explicitly. Yeah. And then you can embed it into the human brain. And here you have a simulation left where the CA1 is on high resolution but isolated. And here it's embedded in the virtual brain. Same parameter settings, but here context matters. It actually changes the propagation patterns and behavior in the simulation. Yeah. So this gives you an example of how you can traverse multiscales. When Egidio spoke about mean field, you can do this mathematically and then stay on one scale, but use this information that traverses the scale. Or numerically, you can also run this type of simulations simultaneously using the co-simulation technology. As a last aspect I wanted to highlight is the following. Yeah, how to use this inference tool. Yeah, when you want to identify an underlying cause. Where is the pathophysiological region? Where is the lesion? If the lesions are invisible in the MRI, or where's the epileptogenic zone in epilepsy? So do I have, when analyzing the functional data, do I have some pathophysiological causes somewhere? What is the nature? Once you can parameterize them, again, parameters are the interface between data, empirical world, and the virtual representation. If we can parameterize it, we can actually interrogate the system through inference. And the inference is after you build the virtual brain twin and you have your hypothesis is parameterized, you can actually introduce a functional data and start sampling and start interrogating. As a scientist, you have to have a hypothesis that expresses as a data feature. If you believe that body temperature is important, you put in a thermometer. You don't do this if you don't think body temperature is a good indicator. So there is a data feature. Here in this epilepsy example, it's actually the envelope function of the seizure propagation. Yeah. And this is what you see here. 3 different seizures, human seizures. So this is also a human seizure where the seizure onset is here. And you here see frequency, time, so the spiking, pre-ictal behavior, and then the seizure onset and the evolution that you see here. And if you unfold it and go in the state space, this is actually the geometry of epilepsy. It's a rest state and the spiral discharge. So in our hands, for this example, the envelope function was the most promising feature. And then you can actually sample the parameters. And here you see actually a real world example. There's the excitability for one brain region. And with the techniques, there are different techniques. These are Monte Carlo Markov chain techniques, MCMC, and they are difficult to use. And we do our best to not optimize, but to make them accessible and as stable and robust as possible, this type of techniques. So this you'll find on the EBRAINS website under validation and inference. This here, the probability distribution of the excitability. Here you have it in a higher dimensional space. So when I talk about sampling, we are sampling this landscape and try to uncover them. Yeah. And then when we communicate with a clinician, this is what how we communicate. Often we provide just a heat map, as you see here, overlaid over the post-surgical MRI, because the epileptogenic zone is a target for the surgery. Yeah. Here's just to give you a feeling of this is how we communicate with the clinician. This is a clinical report per brain region. You see the posterior probability distributions. So this workflow is also available in eBrains. And what I just wanted to show you is one key effect here. When the patients are seizure-free, when you compare the clinical hypothesis with what came out of the virtual brain, here for precision or AUC, as different statistical metrics provide very similar results, you see it corresponds quite well, but it drops down significantly for the not seizure-free patients. And I want to give you a feeling for where this is coming from, because this explains actually some of the power of modeling in the context of data-driven approaches and the fusion within a digital twin framework. Put yourself in the shoes of a clinician. She has a patient, has to make a decision about the clinical implantation of the SEEG. Then based on this, a decision is being made about the organization of the epileptogenic zone. What we do with a digital twin, we model the entire brain. We sample only a functional activity here, but in the inference framework, we provide our knowledge about connectivity, all patient-specific, plus the sample data, and we compute the evidence to how much does it make sense that two confronted to each other, and that provides us with the capacity to make statements about epileptogenic zone also outside of this area that corresponds to the data space. Yeah? And this is actually what provides the predictive power of the virtual brain. And that's what I just showed you, because when you look at the correspondence of the seizure-free patients, when the correspondence is very high, 100%, patients are seizure-free, but it drops down significantly when they are not seizure-free. But the key is that 29% of all these cases, the predicted epileptogenic zone was outside of the accessible range for the clinician. So they were here. And that's the last thought I want to share with you. What we do in eBrains, what interoperation means is you generate a productive, you shed light on a productive relationship between the constituents that you have, yeah, and the hypothesized causes. What we do here is we have data for this particular space. We put it in the context of a virtual brain which allows us a causal extension beyond the data space, and that allows us to make predictions outside of the area that has been sampled, thanks to the fusion that came out of this. And there are other examples in many other domains. Here it's intuitive, this causal extension, yeah, but it is informational, yeah. So let me finish here at this point. If you're interested, there is a summary of these in principles and operations of virtual brain twins. And I thank you for your attention. And I think we still have time for some questions and hopefully some answers. Yeah. Okay. For Egidio, we start with Egidio. A question for Professor Egidio. How exactly does a robot adapt to the extra workload automatically? Is this adaptation driven by the embedded SNN taking the current sensory and error states as inputs? Since the mechanism resembles a traditional closed-loop control system, I'd like to understand what is a specific advantage of using this multiscale SNN controller over traditional control methods. Yeah?
Okay, this is a very good question that often comes when I present the robot that is self-adapting. So the spiking neural network embeds rules that come directly from biological knowledge. So at the beginning, the robot was constructed to understand biology, to understand the implications of these biological rules. And then we discovered that it behaved very nicely and was self-adaptive, was also self-rescaling, which means it can maintain the functionality without saturating top or bottom when the signal is too big or too small. And also can accelerate learning to biological rate through the time constants of learning distributed in different synapses. So one can say at some point we may be able to replace the spiking neural network with an artificial neural network of a different type or with an algorithm. Why not? So somebody will be able to do that. But for the moment, we're demonstrating the principle that with this spiking neural network, embedding plasticity rules distributed in different parts of the network, we can replicate the behavior, the adaptive behavior of a biological system. So that's the point. I hope it was clear enough.
Thank you, Egidio. It was clear to me. Let's see for other questions. I'm switching. Evan has split them between Egidio and Victor questions. So the next one would be, I believe for me, it's in my section, but I think we can answer it both. How can this approach and model contribute to AI? Yeah. So I can start with regard to this. What I have been talking about with regard to virtual brain twins or digital twins, these are models that are mechanistically based. Yeah, they are data informed, but they are not data driven. Yeah, which means they are not limited to the data space, but they can go beyond that. Yeah, knowledge. And that's the reason why I'm always citing Sidney Brenner. Data is not knowledge. But the current AI, if I interpret the question correctly, if we think about foundation models, yeah, you train foundation models, yeah, and what they do is they extract the enormous structure that is in the data. So it is limited through the data. When you link it to additional knowledge, and that's what we are doing with digital twins, yeah, you can actually extend the capacities thereof. And I think this is where the future lies, is in these type of hybrid approaches, yeah, that we combine the deep learning methods, foundation models on the one hand side, with the mechanistic interpretations and realizations that we have in order to generate new knowledge. Would you like to add to this, Egidio?
No, I think this is a clear statement. What I can say is that by design, the virtual brain models and also the spiking neural networks that we embed into robots or we, let's say, use independently in isolation are not designed in principle to carry out artificial intelligence as it is understood now. It is known now, but transformed in the proper way, they may be able of some intelligent performance. So at the moment, we didn't design them to do that.
Yeah. There is one for you. Egidio, can you tell us more about how you integrate connectome simulations with neurorobotics?
Right. So yes, I didn't show very well the scheme of the controller. Connectomics is used to generate, to identify and connect the areas that are most critical for a certain behavior. When you generate a controller, obviously you need to make a choice. The controller is designed to do something in particular. For example, sensory motor coordination or sensory motor adaptation. Okay, and then you have to include the areas inspired by neuroanatomy and neurophysiology that you need to do that. And these areas then are transformed into functionalities. The functionalities can be equations, can be algorithms, or can be spiking neural networks. So we have, first of all, a design of the controller, and then we place a spiking neural network in proper location, and we embedded the spiking neural network with specific functionalities. So the connectome is the backbone to generate the controller architecture.
Thank you, Egidio. Next one for me. Would you speak a bit about solutions in such complex nonlinear models generally? How often do you find unique solutions? Can multiple parameter combinations lead to similar or the same results? Yeah. So the answer is yes, this is something that we are struggling with systematically. It is actually the nature of a biological a multiscale system. You can actually show this mathematically. Every multiscale complex system has the capacity for degeneracy, which means there are not unique solutions. Yeah. One, due to the nonlinearity, one single parameter setting can generate multiple paths. That is the nature of nonlinearity. The nature of the degeneracy is you can have multiple different parameter settings that give rise to the same type of behavior. This is extremely important because this generates actually our type of adaptability and resilience to diseases, for instance. And this, I'm not sure if you have seen it because I had to go fast. That's the reason why we're insisting so much on inference in the EBRAINS environment because of this degeneracy. There is not just one optimal solution, but you have to sample a parameter distribution because there may be multiple solutions to the same question. As a clinician or as a scientist, you want to know this. You want to know these type of realizations. Yeah? And in this one example I showed you with epilepsy, there were multiple answers for the same patient. So it's actually very important. Egidio, if you allow, there is a second question. I'm not sure if it's from the same person, which is linked to this. Do you think this can help to understand heterogeneous individual mechanisms, patient-specific trajectories to similar disease states? I'm citing this because it's very, very closely linked to the previous question. And the answer is yes. Yeah. There is some individual predictive power, despite all of these false positives in tractography, in imaging, and all of these aspects of non-identifiability. You can actually demonstrate that individual data sets provide predictive power for an individual. But of course, you have to be patient and modest. There is a probability distribution with many different realizations, but it's better than using a generic brain twin model. And this has been demonstrated many different times. Yeah. So these are my answers with regard to this. Yeah.
So I wanted to add to the answer from Victor. There are several demonstrations of this remapping of parameters onto single subjects, which means the predictive capability of the model with respect to single subjects. No? So
Yeah.
one clear example is this capacity of the model of predicting the electrodynamics of the entire brain during a seizure that Victor
Mhm.
showed. There is another example. When you run the simulation on a group of subjects, which are inherently different, so there are no humans like the other, they're all different in some detail. And also the pathology, despite the clinical classification and identification of the pathology, they are not identical. So there is a gradation of parameter changes inside the brain of the subjects, no? And actually, what the models are demonstrating, the virtual brain twins are demonstrating, is to be able to capture this differentiation in parameters between subjects. And if you make a correlation between the neuropsychological scores or the neuromotor or scores of subjects and the parameters of the model, you discover that over a certain population of subjects, we have a strong correlation of behavior, which means symptoms or scores, with the parameters of the model, which means the model is capturing with high precision the inherent properties of the networks of individual subjects. So I
Mhm.
think this is a very important point.
Good. Uh, Thank you, Egidio, for emphasizing this. Um, I'm getting more and more questions in, but I'm also being time conscious. I suggest we move. We are still address one or two questions, but uh, one for you. At least it's my section here for you. Could virtual twins be used to simulate task-dependent brain states, for example, during auditory perception?
That's for me or for you, Victor?
It is in the section for you when Evan sent this to me, but I can also, as you wish, I can address
Well,
it
we can do. I
also.
mean, I can start. You can continue. I mean, yes, in principle, yes, in principle, yes. So the brain states are critical for understanding how the twin works. So what basically the twin is doing at the moment is to simulate the brain activity of a subject, which means is imitating the state of the subject at that moment, in that particular moment in this life of the subject. But clearly the brain state can change. Suppose that at some point the subject sleeps, or the subject is excited, or is meditating. Clearly the brain state changes. In principle, the twin is able to capture the changes in the state of activity of the brain.
Mhm.
What would be very nice to have at some point is to have a twin that can change itself, the state, depending on certain parameters. For example, dopaminergic activity rather than serotonergic activity, which is something that is ongoing and would be critical to understand not only the brain states, but also the alterations of brain states during pathologies.
Mhm.
Probably, Victor, you can tell more.
Um, yes, I can address this in a little more detail. Allow me, I'm just reading the questions. I'm getting them specifically from Evan, but I see also that there is a large chat questions. Let me read them. There are two questions from Purgita and Nassim, because I think they are also linked to this. Let's have a quick look at them. What are the present, because they are linked to limitations. Yeah. And the last question can be interpreted in terms of limitations or capacities. Purgita is asking, what are presently known limits of disease simulation based on brain models? Which conditions can be captured? What cannot be captured? Yeah. And Nassim follows up on this. Could you briefly go into the limitations of virtual brain twins? Perhaps limitations that are not obvious and that one would be prone to assume possible with the current technology and methods in eBrains. So I'd like to capture this within integrated answer. Um, Please recognize that the limits of virtual brain twins are defined by, first of all, what we are capturing well is brain activity propagation. We're not capturing brain function in the cognitive sense. Yeah? We are capturing brain activity. We're putting in structural constraints, connectivity. Yeah, we're putting in the physiological constraints, so neural mass models at a particular location. Yes, and then the balancing of this is generates the evolution of the brain activity. Yeah, this is one limitation, and I will tell in a second what we can do with this. Another limitation, of course, is the limitation of our knowledge. Yeah, we do not have perfect knowledge of every single mechanism, and not every single mechanism is being represented in the modeling. So there are aspects missing. Yeah, and especially in the context of inference, when you have the sampler, this inference machinery, yeah, that is trying to balance the evidence captured by the data, the empirical data, and then the explanatory power coming from the model. If a mechanism is missing, yeah, the sampler has to somehow do something, yeah. It either fails, it provides us with a flat probability distribution function. That would be great, yeah, but because then it's uniform, which means we don't have clarity about what parameter fits best, because each model, the mechanism is missing, is equally bad at explaining the data. That is one possibility. Other possibilities are that simply the sampling fails, etc. So these are our limitation of our knowledge as it is implemented causally in the models is certainly a limitation. Yeah, but we cannot implement everything, because then the parameter space becomes very huge. So that's the reason why we need smart, intelligent questions, very targeted specifically to the individual applications. And this is where we stand now with the virtual brain twins today. They are wonderful to guide development of new technologies, such as neurostimulation. That's one possibility, where we are applying them. Pierpaolo Sorrentino, for instance, is applying them in the context of multiple sclerosis or in Parkinson's disease, or the Charité lab around Petra Ritter is also looking for improving the stimulation effects in invasive stimulation using Parkinson's disease. So questions where we are linking physics, so the mapping between sources to sensors that we control quite well, and where we have a decent understanding of the underlying pathophysiology, putting this all together in a shared ecosystem. This is where the virtual brain twins shine. Other applications is healthy aging, for instance. Healthy aging, other neurodegenerative diseases where consequences express themselves in the functional brain imaging data. So these are good playgrounds. Where it often fails is when you go across multiple scales and there are too many mechanisms, too many possibilities, degeneracy. So there it's a little complicated. Yeah. Okay, sorry, I tried to put this under one umbrella. Shall we see if there is still something? I'm looking at the questions, everyone. Last question, and Egidio, maybe we can answer this together, and then I think we need to liberate people. Last question. Could the virtual brain be extended to simulate lamina-dependent signals by incorporating lamina-specific neural mass models together with a laminar neurovascular forward model, such that model predictions can be directly compared with experimental laminar fMRI data? I have an answer. Do you want to? Egidio first?
I can start. Yes, I can start. I mean, in principle, the answer is in principle, yes. Depends on the fine grain that we put into the representation of the structures of the brain. So if the representation is good enough, the answer is yes, we can. So the process of technological development of the virtual brains is evolving. And with the technology of the mean field models, we can actually represent the different lamina. So we can actually represent the problem in mechanistic terms and then simulate it. So this is a very simple answer, but it means yes, we can. Probably at the moment we can't do it yet, but in the future, with the development and the implementation of the mean-field models at high resolution, we will be able to do this.
Mm-hmm. Yeah. And my answer, so this was Giuseppe's question. Giuseppe, thank you for this question. You're actually hitting a very important point, in my opinion. Laminar dependent signals, and of course, the laminar specific neural mass models, this already exists to some degree, but the linkage to neurovascular models, yeah, this has not been established. There are two aspects, yeah. So astrocytes on the one hand side, yeah, and then vascularity on the other hand. These are two elements that need to be integrated. With regard to glia and astrocytes, Maria-Lena Lenne has generated lovely neural and glial models. We are in the process of looking at those and to implement them in the virtual brain, but it has not happened. So it's ongoing at the moment. But the vascularity, we have treated this extremely naively so far, and there are loads of data available, and it falls beautifully into this into the forces of what we have in eBrains, namely interoperating different data, heterogeneous data that need to be co-registered in the same ecosystem, and then physiologically through mathematics connected, namely neurovascular couplings, but here with a spatial structure. This is head-on this question. It does not exist. It's prioritized, at least in my thinking. As a community, we need to find people that wish to go into this particular direction, provide data, provide the mathematics, do this together, and it will open up completely new avenues, such as in stroke, for instance, yeah, or
Absolutely.
better interpretation of perfusion and so on. Yeah?
Absolutely, because this is a very critical point. I said the last word on this, because different parts of the brain have different hemodynamic response functions, but at the moment we use the same for everywhere. No. The other thing is that there are different mechanisms now for neurovascular coupling that we can't apply everywhere. We have to be specific. At the moment we can't, but we know it should be done. But with a mean-field model in which you have a specific representation of the population of neurons and of the inherent mechanisms of the network, then you can do it. Then you can do it. And for the case of the cerebellum, we've done it. And the neurovascular coupling is now based on the population of neurons that do it, not on the others that don't do anything today. So we are able to dissect the system. The better we represent it, the better we will be able to use it.